Welcome to the Basics of Animal Development module, and in this talk we're going to learn about fate maps. We're going to first define the term morphogenesis, which is an important feature of development. We will then look at the main types of cells in a developing embryo and list different cellular processes that affect embryogenesis. We will then explain the concept of a fate map and look at different ways that are used to determine the fate maps of developing organisms. Many different processes affect embryogenesis, and morphogenesis is the process that causes the organism to develop its shapes. For morphogenesis to occur, the cells of the embryo have to undergo different changes. But for that, let's first look at what are the different types of cells in an embryo. There are two main cell types in an embryo, and the first one are the epithelial cells. The epithelial cells are cells that are tightly connected and can form structures like sheets or tubes. The second type of cell that's normally found in an embryo are the mesenchymal cells. Mesenchymal cells are not connected to each other, or if they are loosely connected to one another. Now, what should be remembered is that epithelial cells can become mesenchymal cells through a process called as epithelial mesenchymal transition. Similarly, mesenchymal cells can become epithelial cells through a process called as mesenchymal epithelial transition. In order for development to occur, the cells can affect the migration patterns, cell growth, and cell division of neighboring cells. Thus, cellular processes are present that can affect the process of morphogenesis. The different processes that affect morphogenesis are the direction of cell division as well as the number of cell divisions, changes that can happen in cell shape, the migration of cells from one region to another, cell growth, cell death, changes in composition of the cell membrane or its secreted products. All these cellular processes play a role in morphogenesis. We're now going to look at what are called as fate maps. Early scientists who studied embryology always wondered the fate of cells during the process of development. Basically, in other words, we're trying to say, how do we know what kind of cells develop into what kind of organs, for example? So, through many different experimental procedures, scientists were able to study the fate of the cells and were able to generate maps called fate maps that basically tell us what part of a zygote or an early embryo forms what part of the embryo. So in the picture to the right, where we see the embryos of zebrafish, frog, mouse, and chick, they're all in the gastrulation phase, and the different colors tell us which part of the embryo forms what part of the organism. For example, the blue parts can form the epidermal ectoderm in the case of the zebrafish. The red part forms the mesoderm. The dark red forms the notochord, and then the light yellow forms the endoderm. In the case of a frog, in addition to the epidermal ectoderm, the mesoderm, the notochord, and the endoderm, we also have the neural ectoderm that's shown right below the blue. Similar fate maps have been generated for mice and chick embryos. One way to study the development of cells in an embryo is through the direct observation of cells that have a different color from other cells in that embryo. This can be observed in a very few organisms like that of the tunicate. As shown in figure A, in the case of the tunicate, few cells of the early embryo actually have a naturally darker yellow color. And as we can see in this 8-cell stage, we have 2 cells that show this dark yellow color. What scientists did was they looked at, as the development occurred, how that yellow colored cell was able to determine its fate. And so what we see in figure C, in the case of the yellow colored cell, it actually became muscle cells of the tunicate. Thus, by directly observing this cell that had a different color, scientists were able to determine the fate of that cell during the process of embryogenesis. To ensure whether these yellow-colored cells genuinely formed muscle, scientists removed these cells during the early embryo and looked at what kind of larvae did these specific embryos produce, which did not have those yellow-colored cells. And what they found was these larvae, where these yellow-colored cells were no longer present, they basically had no tail muscles, thereby confirming the fate map of these cells. However, not all organisms show different colored cells. And hence, scientists decided to use vital dyes, which are basically dyes that can be observed through the naked eye because they have a color associated with them. These vital dyes can be applied to specific areas of the embryo to stain certain types of cells, as shown in the figure below. The stained cells can then be followed over time to determine what happens to those cells and what kinds of organs do they form. However, one drawback of this method is as the cell undergoes cell division, the intensity of the dye becomes diluted, and hence sometimes it would be harder to follow the fate of the cells towards later embryogenesis. To bypass this disadvantage of vital dyes, scientists decided to use a different type of dye, which was the fluorescent dyes. Fluorescent dyes are much brighter than vital dyes and can be injected into the single cells of an embryo. These single cells can then be monitored during the process of development to determine what cell lineages they form, because these cell lineages remain fluorescent after many cell divisions, as shown in the figure below. Another way to study development is through genetic labeling. In the case of genetic labeling, forming chimeric embryos was a popular method. Chimeric embryos are made from tissues of different genetic but similar sources. For example, a quail and a chick. They are genetically different, but they also show a lot of similarities. And hence, chimeras were made where the embryonic quail cells were grafted into that of a chick embryo, as shown in the figure below. We can then map the fate of these cells by seeing what part of the chick has quail cells. If we look at the figure to the right, the part of the chick that actually has the dark coloration basically came from the quail. And this way we note that the cells that were chosen from the quail to form the quail-chick chimeras basically ended up becoming the wing of the chick. Since it's not easy to make chimeric embryos of all kinds of organisms, transgenic DNA chimeras have also been a popular tool. In this case, a transgene, or basically a piece of DNA that codes for a protein like the green fluorescent protein or GFP, is inserted into the cells during the early embryonic stages. These transgenic cells can then be grafted into a non-transgenic embryo from the same organism. The transgene will then express its protein, which, for example, is the green fluorescent protein that will illuminate the transgenic cells as they migrate and develop into the tissues and organs of the embryo. This method can produce very precise fate maps as we're able to observe the fate of these transgenic cells during the process of embryogenesis. Thus, we have seen different tools that have been available to study developmental biology. With this, we come to the end of the talk where we learned about morphogenesis and the main types of cells in an embryo, which were the epithelial and mesenchymal cells. We also learned about different cellular processes that affect embryogenesis, as well as different ways to determine fate maps of a developing embryo.